I'm Ryan Urbanowitz, and this is part 2 of Machine Learning Essentials for Biomedical Data Science. In this video, we'll discuss biomedical data and the respective goals and challenges therein. Here's a quick reminder of the parts making up this video series. In the last video, we covered some basic terms and concepts related to machine learning. Diving in, anyone who's worked with biomedical data will probably tell you that it can be a significant challenge. First off, raw biomedical data can come from many sources and in many forms. For example, it could come as clinical notes, test reports, electronic health records, or photographs such as X-rays or MRI scans. Some data may already be in a structured and digitally accessible form, for example in a database. But many sources of biomedical data start in an unstructured and non-digital form. Such data must first be pre-processed and/or wrangled into a structured format prior to conducting any kind of statistical or machine learning analysis. Let's review some common data types. First, we have tabular data. This is simply the format most of us would be used to, involving rows of instances and columns of features. Next, we have a directed graph or a network. For example, this is often how ontologies of terms are arranged. Then, of course, we could have images or videos, which are simply images over time. Quantitative data can also be measured over time, which we refer to as time series data. Examples of this might include patient heart rate or electrocardiogram measurements over some period of time. And our last example is text-based data, which is often unstructured and might require the application of natural language processing techniques to extract useful, structured information. Most data sources could be extracted into a form of tabular data, which is the most conventional format to use with machine learning. To that end, the concept of feature extraction is a topic we'll cover further in the next video. Here on out, we'll be primarily thinking of tabular data when we discuss using data. One important consideration for data science is the variable types. Understanding variable types is important for selecting the right analysis methods and ensuring downstream machine learning treats respective variables appropriately. Let's quickly review different variable types. This is applicable both to feature and outcome variables. First off, a variable can be numeric, otherwise known as quantitative. For example, this could be the age of a patient. Numerical values might be continuous valued, appearing anywhere within a numerical spectrum, or they could be discrete, for example, only appearing as integer values. Going back to this age example, we know that true age is a continuous value. However, in patient data, age is usually captured as a discrete integer. Next, a variable could be categorical, otherwise known as a qualitative variable. For example, this could be something like patient race or biological sex. These variables have no intrinsic ordering to them. Of note, for many machine learning algorithms, categorical variables as features present an extra challenge. Specifically, most algorithms train models under the assumption that all variables with more than 3 possible values have some quantitative ordering. One way around this issue is to re-encode categorical features using something like one-hot encoding, which breaks a single categorical feature into a set of new features, one for each possible category. So for example, if our categories were different colors, we'll have one binary variable that asks whether the color is blue or not blue, another binary variable asking whether the color is red or not red, and so on and so forth. And lastly, a variable can be ordinal. For example, this could be something like tumor stage. These variables are similar to categorical in that they often appear in data as text entries, such as stage 1 or stage 4. However, these values can be re-encoded as numerical values with a quantitative ordering. Notably, there are generally some assumptions that are being made when we numerically encode ordinal variables. For instance, how far apart are the different groups? After encoding, these variables can be treated as discrete numerical variables. Also of note is that discrete variables with only 2 values are referred to as binary, and we generally encode these with 0 or 1 values. Categorical variables with only 2 values are typically called Boolean variables, described with the values true or false. Here we see the results of a COVID-19 test, which might be labeled as positive or negative in a given dataset. It could also be labeled as true or false, or encoded with a 0 or 1. variables with only two possible values are easier to deal with since the order between them is pretty much arbitrary. The only consideration might be to ensure that all variables in your dataset have a numerical encoding if the algorithm implementation requires it as part of the data formatting. Having reviewed feature types, let's look at an example of some tabular data. So here's our dataset. The rows represent individual instances with a header at the top. The header identifies the different columns in the data, or in other words, the names of the different variables in the data. Included in these columns are feature variables, also the outcome that our downstream model will aim to make predictions on. And sometimes the data might include an ID, which identifies the unique instances of the dataset. Of note, instance IDs should not be provided as a feature for machine learning modeling, as it is a unique value for each row and thus offers no information for the generalization process. Now let's identify some of the unique variable types we discussed in the last slide. The feature age is an example of a discrete variable. Both zip code and biological sex are examples of categorical variables. Body mass index, or BMI, is an example of a continuous variable. And income, as presented in this dataset, is an example of an ordinal variable that also have a known ordering, in this case low, middle, high. Depending on the algorithm implementation to be used downstream, we might want to re-encode the values of income with something like the values 0, 1, or 2, corresponding to low, medium, and high. And lastly, we have the feature insured and the outcome diagnosis. Both of these could appear as Boolean or numerical based on how the values were recorded or whether they were encoded as a 0 or 1. Modern-day biomedical data has a tendency to be big or have a very large number of features and possibly instances. As we alluded, biomedical data can come from many sources, including features and other data streams from the genome, epigenome, proteome, microbiome, microRNAome, electronic health records, imaging, mobile health, and many others. In trying to understand and predict biomedical outcomes, it may be desirable to integrate all available data into a single dataset for analysis. However, this comes with a number of logistical concerns like data storage and computer memory limitations, as well as analysis concerns, such as the efficacy of feature selection and modeling in the context of having a huge number of potentially predictive features. Now let's take a look at some of the typical goals of machine learning when applied to biomedical or clinical data. First, we might want to identify risk factors or biomarkers of health outcomes. This might be in order to understand disease etiology, in other words, the cause of disease, to make diagnoses or other outcome predictions using those risk factors, or to improve personalized medicine by predicting who would benefit the most from preventative medicine or what drug or treatment to administer, if any. We also might want to develop an outcome prediction model for deployment, for example, in a clinical setting. The goal here is typically to make more accurate predictions, or simply to improve clinical efficiency by either increasing the speed of diagnosis and treatment or reducing costs. Whatever the goal, in the simplest framing, machine learning starts with data, which is processed and passed to a machine learning algorithm, which in turn trains a model, and this model can be used to try and understand the relationship between features and outcome, as well as to make predictions on future data instances. Next, let's review some of the common challenges we might need to consider or overcome when conducting a biomedical data analysis. I've separated them here into some general categories. First, we have cleaning and data preprocessing. Here we may have to deal with handling missing data values, diverse feature types, and preparing features for modeling with feature engineering. When it comes to analysis, if we're doing classification, we'll need to worry about class imbalance, which is when we have many more of one class than another. Class imbalance can impact model training, as well as require us to be careful to take that imbalance into account when we evaluate our models. Additionally, we should be aware of covariates in the data. These should be taken into consideration when using machine learning as a vehicle to try and better understand the potential causes behind outcomes, rather than only being concerned with making accurate predictions. Next, we have computing. The main concern is the scale of the data, which is based on the number of features or instances in the data. Then we have challenges with respect to signal detection. For example, data can be noisy, which means that even with an optimally trained model, we can never expect to make 100% accurate predictions. On the other hand, it's possible that some, or perhaps even all features required to understand an outcome could be missing from the data. For example, if you're failing to train a highly predictive model of lung cancer when genetic data is available, but environmental data, such as pack years of smoking, is not. Additionally, it can be difficult to model the impact of features with rare values. This is common, for example, in genomic data, such as whole genome sequencing data or whole exome sequencing data. And lastly, in signal detection, the relationship between features and outcome can be obscured by complex multivariate associations, which we'll discuss in more depth in the next couple of slides. And lastly, we have challenges in data and model reliability. Here, we concern ourselves with the reproducibility of our results on other future data or data from another hospital, region, state, or even country. Trusting a model is often paired with the challenge of being able to interpret or explain the predictions of a model. And lastly, we want to identify and account for the possibility that our data or model may have underlying bias. Now let's take a closer look at the challenge of complex patterns of association in data. We'll do this from the perspective of genetic data. First, here's a schematic attempting to illustrate some of the possible ways that features on the left can be associated with outcome or phenotype on the right. Here, the word phenotype just refers to some trait of an organism. Over the next few slides, we're going to use phenotype and outcome interchangeably. Features here could be any of the things we've already mentioned. At the top, we have the simplest association between a single gene or genetic feature and the target outcome. This is known as a univariate association. Next, we have pleiotropy, which is when a single gene or genetic feature has an association with two or more seemingly unrelated outcomes. Next, we have epistasis, also known as a multivariate interaction. Here, two or more features interact with one another to impact outcome. Notably, in the most extreme case, it's possible for feature interactions to strongly predict outcome even when none of those individual features has any univariate association. And lastly, we have heterogeneous associations. The first heterogeneous association we'll mention is genetic heterogeneity. This is a situation where different genes or genetic features can produce the same or similar outcome in different instances. Phenocopy is a similar situation where an environmental feature could impact the outcome of instances in the same way as a gene or genetic feature. And beyond any of these individual, more complicated associations, in real life, they might occur in some additionally complex combined manner. Understanding that complex underlying associations may exist in your data is an important part of how you go about key elements of a biomedical data analysis pipeline, so that you don't miss these possible relationships. Now let's focus a bit more on the unique challenge presented by epistatic interactions. First off, note that there can be a difference between biological and statistical epistasis. Biological epistasis implies that there's a physical, pathway-based, or causal interaction with respect to outcome. Statistical epistasis is limited to an interaction between features that are predictive of outcome. This might happen independent of a biological interaction. However, we want to use statistical or machine learning methods that are capable of detecting statistical interactions that still may be essential to training an optimal model. Next, let's contrast multivariate effects in general with those that would be considered multivariate interactions. Let's start by looking at a simple multivariate additive effect, where each feature contributes its own univariate effect that adds up in impacting phenotype or outcome. In this illustration, we have two features, each with only two possible values, 0 or 1. Feature 1, in green, has its values represented in the plot by the colors red and black. And feature 2, in blue, has its values represented in the plot along the x-axis. The y-axis is the phenotype or outcome which we're trying to predict. The 4 dots on the plot represent the phenotype value, let's say something like patient height, given the corresponding values of feature 1 and feature 2. So as a baseline, imagine that if neither feature had a relationship with phenotype, the phenotype would be the same for all the points. First, we can visualize the univariate effect of feature 1 on phenotype as the difference of the average of the red dots versus the black dots. Next, we can visualize the univariate effect of feature 2 on phenotype as the difference in the average of the left-sided dots from those on the right. What should be clear here is that both features have their own independent association with phenotype, and that these effects add up, leading to a larger value of phenotype cumulatively. However, there is no interaction effect here. Now we'll look at a multivariate multiplicative effect. Again, we have the same setup, where we can confirm that both feature 1 and feature 2 each have their own univariate association with outcome. Here's feature 1, and we can see the big difference in phenotype, and feature 2, again with a difference in phenotype. However, overall, we can see that the combined effects of the 2 features on outcome leads to a greater change than the sum of the 2 individual effects. Therefore, this is considered to be an interaction effect. The impact of interactions on outcome can only be fully appreciated when the values of the features are considered together. Lastly, we look at a multivariate crossover effect, also referred to as a masking effect. Here, the effect of one feature on outcome can be partially or fully hidden by another. Using our same example, we can see that for feature 1, the average phenotype of red versus black dots is almost exactly the same. So if we had used a univariate test of association for feature 1, we might incorrectly think that feature 1 is irrelevant to predicting outcome. Looking at feature 2, we see an extremely small univariate effect, which we might not be able to distinguish as interesting from other noisy features in a given dataset. However, what should be evident from these orange circles is that despite outcome having no clear relationship with either individual feature, the specific combination of feature 1 and feature 2 values very much impacts whether the instance has a high or low phenotype value in the data. While we don't know ahead of time the true nature of complex multivariate associations going from one dataset to the next, it's important to consider these worst-case scenarios of pattern complexity when deciding what assumptions you're willing to make in setting up your data analysis pipeline. Next, let's dive a little deeper into how complex associations such as epistasis can make data analysis and machine learning more challenging. However, before we do that, let's talk briefly about what a search space is. This knowledge will be useful well beyond our discussion of epistasis. A search space is a set or domain through which an algorithm searches for a solution, for example, searching through different model configurations. A search space might be a well-defined and finite data structure, like searching a database for matching patient IDs. Or it could be a vast and possibly infinite set, like a very flexible machine learning algorithm searching for an optimal model. In general, the search space of modeling is impacted by the number of features, the way an algorithm represents knowledge, and the assumptions that algorithm makes about the data. However, beyond modeling itself, search also plays a role in finding the optimal hyperparameter settings, as well as optimizing feature learning or feature selection. Now, let's combine our discussion of epistasis with what we know about search space to discuss the curse of dimensionality. The curse of dimensionality refers to various phenomena that arise when organizing or analyzing data in high-dimensional spaces that don't occur in low-dimensional ones. For example, analyzing univariate effects of features in a dataset would be low-dimensional, but exploring all unique feature combinations would be a high-dimensional one. First, let's talk about the phenomena of increased computational burden. To put this into context, let's start with a small search example, like the game of tic-tac-toe. If you want to search for an optimal strategy to play this game, how many unique games of tic-tac-toe would you imagine there could be? It might surprise you to learn that there are, in fact, 255,168 possible unique games that could be played, with outcomes of win, lose, or tie. Now consider a larger example, such as conducting an exhaustive analysis of 500,000 genetic variants and associations with outcome in a genome-wide association study, or GWAS. If we only looked at univariate associations, our search space would intuitively be 500,000 possibilities. However, what if we wanted to look at all univariate effects as well as all possible 2- and 3-way interaction effects among those 500,000 features? Now our search space has climbed to approximately 2.08 times 10 to the 16th possibilities, which happens to be 100 million times greater than the number of stars in the sky. Let's generously assume that your personal computer is able to complete 200 million search operations per second. That means that this search would take a minimum of 120 days to complete. Beyond computational burden, the curse of dimensionality also dictates that as we consider higher order combinations of variables, we will observe exponentially fewer instances for each feature value combination. For example, here we see instances separated into quantitative value regions, either as a single feature, a pair of features, or 3 features. Notice that as the dimensionality increases, the number of instances that end up in any partition of the overall problem space becomes more sparse. And this goes to the point where some or many partitions will have no instances in them at all. This phenomena directly impacts the power, which is the probability of a hypothesis test finding an effect if there is one to be found. Essentially, searching in higher dimensions is the same as trying to find an effect with too small of a sample size. The point of this slide is not meant to make you feel hopeless about the challenge here, but rather to make you aware that there are some limitations to applying traditional analytical approaches and machine learning, and that sometimes it's not practical to try and tackle an exhaustive search of the solution space, but instead to more cleverly and efficiently explore a subset of the search space. The last part of this video will cover one of the other complex associations we discussed, specifically heterogeneous associations, such as genetic heterogeneity and phenocopy. To understand this complicating phenomenon further, assume we had a group of patients that all had a disease outcome of interest, and that from a clinical perspective, the disease phenotype or phenotypes of these patients was either similar enough that we couldn't reliably distinguish them, or we observe a range of phenotypes, but stratifying patients by their severity of disease phenotype doesn't help us to better model underlying associations. Either of these scenarios is possible when we're dealing with heterogeneous associations. Imagine that under the surface, these patients belong to underlying subgroups, each with a unique set of features that predicted or caused their disease phenotype. So for example, the outcome of these patients are best predicted by these 2 genetic variables, but this subgroup of patients is best predicted by this environmental variable, and so on. Heterogeneous associations are a likely contributor to missing heritability, or unexplained genetic risk of disease, and is likely an important part of developing more effective personalized medicine. From a machine learning modeling perspective, genetic heterogeneity can be a difficult problem to overcome, since ideally most algorithms seek to train a model that can be uniformly applied to predict outcome of all instances. One of the reasons I've included rule-based machine learning algorithms in part 7 of this video series is that those algorithms have been demonstrated to effectively detect and model both epistatic and heterogeneous patterns of association. This is in large part due to their ability to break problems down into a piecewise model of individual if-then rules. Our last slide dives a bit deeper into a specific example of heterogeneity and its relationship with improving personalized medicine. This example was taken from a book chapter by Kumar in 2016. Here we have a group of patients with adenocarcinoma of the lung, otherwise known as lung cancer. The different color of each patient reflects heterogeneity according to respective driver oncogene mutations they each possess. For example, EGRF is an oncogene that is known to be a predictor of response to EGRF TKI, a tyrosine kinase inhibitor applied as a treatment. The number of people in each heterogeneous group reflects the approximate incidence of each oncogene in the patient population. 90% of EGRF mutations, including L858R and exon 19del, are known to be predictors of a desirable treatment response. However, there is further heterogeneity in patients with specific EGRF mutations, and these patients had a varied response to EGFR TKI as a treatment. We can see here some other heterogeneous predictors of inherent resistance to the treatment, leading to shorter survival outcomes. And beyond the patient group as a whole, there's further heterogeneity in individual patients treated using targeted therapy that results from the selection pressure and microenvironment of the tumor. So in this case, treatment can actually exacerbate the heterogeneity of tumors in the patient. Stepping back, this example is further meant to emphasize the unique challenges of biomedical data problems. They can be noisy, large-scale, complex, and have lives on the line when it comes to decision-making. This arguably makes biomedical data science one of the most challenging, interesting, yet rewarding places to be a practitioner of machine learning and data science in general. Thank you for joining me for part 2 of Machine Learning Essentials for Biomedical Data Science. In the next video, we're going to cover the preparation of data for machine learning. If you enjoyed this video, please like and subscribe to follow our future content.